Welcome back to MLT 120. Today we're going to learn about the Wright stain. We're going to learn about some principles and what is in the Wright stain and how to perform a Wright stain. The Wright stain is referred to as a polychromatic stain, meaning that the Wright stain is, or it stains different parts of the cell different colors, so polychromatic. The specimens that we're going to use to make our Wright stains are well-mixed EDTA tubes. These are from venipunctures, but they also could be from capillary punctures. And we're going to take those EDTA samples and we're going to make a blood smear on a clean glass slide. The fixative that's usually used for a Wright stain is methanol. And this means it fixes the cells to the slide so the blood cells do not wash off during the staining process. There are two stains contained within the Wright stain, and the first stain is eosin and the second one is methylene blue. Now for the principle for our Wright stain. The eosin is an acidic dye that stains the cytoplasm the pink to reddish color, and it stains the alkaline parts of the cell. So the alkaline parts are being stained with the acidic dye. For example, the hemoglobin in the red blood cells are being stained a pinkish color, and the eosinophils are granulocytes. The granules are being dyed a reddish orange color. The methylene blue is an alkaline dye, and it stains the nucleus and RNA blue. So it's staining the acidic parts of the cell with the alkaline dye. Neutrophils get their name because the granules are neutral in pH, so their granules are going to stain a pinkish purple color in the Wright stain. Now for the manual staining procedure for the Wright stain. First thing you're going to do is you're going to put some Wright stain into Copeland jars, and then you can dip your blood smears into the stain about 5 times 1 second each. Another option would be just putting your smears into the jar and leaving them for a minute, and then you're going to pull your slides out of the stain, and then you're going to allow it to drain a little bit, and then here it says to air dry, but what we're going to do in lab is we're going to transfer it over to a Copeland jar with water in it, or distilled water, and we're going to rinse our slides, and then we're going to allow our slides to dry, and then we're going to observe, and we can observe on 40x, or we can go up to the oil immersion 100x objective on our microscopes to see our cells. There are automated slide stainers available for Wright stains. Many times you'll find these stainers in your higher volume labs. Most of your hospital labs will have a Wright stainer, and your Wright stainers will have or start out with doing the fixative of methanol before they go through the staining procedure and doing the eosin and the methylene blue. But before the slide comes off of the automatic stainer, it will come off dry. So it'll be completely ready to read once it comes off the automated stainers. And these automated stainers just really help turnaround time when you're in a high volume lab, and it cuts down on the time that a tech is standing there at the sink making and staining slides. The tech can just stick the slide on the stainer and it stains while the tech is able to do other tasks throughout the laboratory. This concludes our little lecture section on Wright staining. We will be completing Wright staining in lab. If you have any questions, don't hesitate to reach out.